Okay, so I'm Joanne. I'm going to be presenting with Noah. We're going to be talking about the history of vaccines and inactive vaccines. So this is our topics covered. We're going to be starting off with variolation vaccination from the 1970s and eradication with the 1900s. I know some of these topics are going to be covered by other groups, but we're just introducing it to better explain the history or the timeline of events. And then we're going to end with types of vaccines. So we're going to start off with a brief history, and Noah's going to be covering it.
During relations. So this is when there is a fluid containing virus. So it's lost variants due to an immune response. So then it creates immunity without causing the disease. The first to use this was when they were testing smallpox. So they took material from patients or a recently infected individual that had smallpox in hopes that it would infect other people with a less severe result. So in this case, it did infect people with a much lower fatality rate than normal smallpox infections, but it also spread other diseases such as syphilis and hepatitis. So this was widely practiced in Africa, India, and China in the 1600s. So then vaccinations. So vaccines work by mimicking disease agents and stimulating an immune response to build up a defense against them. So the first is the first step in vaccination would be capturing the antigen. So the antigen presenting cell will roam the body looking for invaders, and then it's going to go to T helper cell activation. So the naive T cell, the naive T specific cell to the antigen will recognize it, that it's a foreign, and become activated. So then the T helper cells will then create a response to kill the invader. And then B cell activation. So the naive B cells will react to the vaccine agent when it enters the body. And then B cell matures into plasma B cell after activation by the vaccine, antigens, and recipients of the signal from the activated T helper cells. Some B cells will transform into plasma B cells. The immune system then will have antibody factors. And then we go into plasma B cell secretes antibodies. So it's a Y-shaped protein called antibodies that release a high levels of every second of secretion. The number of antibody types in the human body is in hundreds. So it's always binding. And then we're going to go to antibody binds to a specific agent. So each antibody is tightly attached to one specific target agent with a similar lock and key method. And then it kills T cells in response. So the vaccine contains attenuation virus. The vaccine virus enters the cell, kills the T cells, and destroys the invader cells. So, and then the last step, so it doesn't happen again, is retention of memory cells. So the goal of the immunization is to produce memory of vaccine antigens. So then we move on into attenuation. So attenuation vaccines are either live attenuated vaccines, so it is created by reducing the virulence of pathogens, but it's still kept alive. So it takes the infection and it alters it to become harmless or less virulent. So attenuated vaccines stimulate a long and effective immune response. It's long lasting and it encourages the body to create antibodies and memory immune cells in response to the specific pathogen. So some examples are measles, mumps, rubella, yellow fever, and some influenza vaccines.
So that takes us to the 1800s with toxoid vaccines. Toxoid vaccines are inactive, or they need to be inactivated before giving it. And in this case, antibodies were actually discovered. So what they did, they took the proteins created after infection, bind it to a pathogen, and that binding would inactivate it. So with the discovery of antibodies became the era of antitoxins. So when scientists discovered it, they wanted to create these antibodies in large batches or large doses so they could administer it to more people. The way they did this was they would inject a virus, in this case tetanus, to a mammal, in this case a horse. The horse would then develop antibodies against it, and scientists would harvest it, purify it, and administer it to patients that were showing symptoms of the illness. However, with this whole step-free process, they actually injected actual tetanus into patients, and that caused a lot of injuries and death. And because of that, the FDA was created, the Food and Drug Administration. Toxoid vaccines in general is going to be covered more in detail by another group. This takes us to the 1930s with the invention of the microscope. So we know today that microscope uses traditional light to be able to visualize and see particles. However, in this case, they used probes, and with this, they discovered virology. So that's when scientists would kind of classify viruses and whatnot, or particles, according to shape and size. And they started to catalog viruses, which created vaccines. With the invention of the microscope, that led to thorough research into the Spanish flu and into poliovirus. And on the figure to your left, you'll see a replica of the first microscope invented. After the invention of the microscope came eradication. So the goal was to eradicate smallpox completely. This is just a picture of the newspaper clipping written by Dr. George. And again, this was between 1950s and 1960s. The goal was to vaccinate everyone. The only exception were if you had medical reasons or like allergies, for example. And if you chose not to be vaccinated, you would have to pay a big fine, and you were banned from attending school and work. However, with this whole eradication process, they were able to diminish smallpox. And it was actually the first human virus to be eradicated. The last case detected was in 1978. So now we're going to move on to inactivated vaccines. We're going to be giving a few examples. So how does it work? You're going to take the inactive vaccine. It's going to be entering the human body, either syringe and needle through the skin or orally through the mouth. It's going to enter, and antigen presenting cells are going to take it, engulf it, chop it up into little pieces. Those little pieces cannot join together to replicate. Once it's chopped up, it's going to move over into the membrane of the cell, where the body is going to recognize it as an antibody or an antigen. To Noah.
So then we see hepatitis. So it is inflammatory of the liver. So vaccines for hepatitis are available in the United States. It took place as a routine immunization in childhood since 1994, since there was an outbreak. And then the hepatitis vaccine is recommended at age 1 with the booster. So if you are traveling, they do recommend hepatitis shot because it can be triggered with a pre-departure hepatitis A vaccine. And then the benefits of a single vaccine administration should last several months, but a second dose will have long-term immunity. And then we have influenza. So influenza is a respiratory illness caused by influenza virus. There are many types of influenza, A, B, and C. There are many strains of each types as well. So 4 to 5 months after the 3 vaccine strains have been selected, so either in June or July, the vaccine strains develop in a separate test for purity and potency, and then it is administered. Influenza is a primarily transmitted via infected respiratory droplets by air, coughing, or wheezing. That is how it's contracted. And then I'll pass it back to Joanne for polio.
Okay, another inactive vaccine is polio. It was invented by John Salk and his team. It was actually really common in the US, killing and paralyzing thousands, roughly 1,500 or 15,000 each year. They're kind of close to ending polio, but it still becomes, or it's still a big problem in Asia and Africa. It's very contagious from person to person. And all children in the US receive the polio vaccine. Because it's inactive, you need a bunch of boosters just because of it's not as strong as the live vaccine, for example. So in polio, their kids are going to receive 4 doses. It's recommended for adults to take the vaccine if they are traveling to Asia and Africa, for example. In the US, they give polio as a shot, depicted up here, and in other countries, they give it orally, depicted down here. Elvis was actually pro-vaccine, pro-polio vaccine. So he went ahead and had this whole thing published in articles and on the news, and it pushed a lot of people to consider and take the polio vaccine. Next up is rabies. Rabies is another inactive vaccine. So thousands die every year. However, it's rare and preventable. In this case, it's passed from animal to human, but it can also pass from animal to animal. So when passed to the human, it's going to inflame our brains. And then when it's passed through animal, it's going to enter the bloodstream through the spinal cord and then into the brain, infecting the whole animal themselves. Those that should not receive the rabies vaccines are patients who are immunocompromised, so that includes AIDS, cancer, those who are pregnant and breastfeeding. With rabies, this was actually common where I'm from, so this was so new for me. We don't have rabies back home, so that's pretty interesting. Other than that concludes our presentation. Here's our references.